This video covers the IGCSE Biology Objective 1.3.7 on the 2025 CAIE syllabus to state the features of viruses. So first of all, let's take a look at virus classification. So as we look at virus classification, viruses are classified or organized into four general categories based on the structure of the virus. So you can see here some of the pictures. So viruses are not part of the kingdom classification system that living organisms are put into. So they're completely separate. It's not even a kingdom, it's just virus classification. So we have helical or spiral shaped viruses. The example that is here is called the tobacco mosaic virus. This is a virus that attacks the tobacco plant. We have polyhedral, that is the shape, a polyhedral shape. The example of this is an adenovirus that would attack living animals. We have spherical viruses that are circular. This is like the flu virus and those sorts of similar type viruses that attack animals. And then we have viruses that are what are called complex viruses, meaning that they have other sorts of structures aside from just the general shape. So this one here is called a bacteriophage, and this one actually specifically attacks bacteria cells. Okay, let us consider if viruses are indeed living organisms. I did previously say that viruses are not even in the classification system that we apply to living organisms. And so as we look at the characteristics of life, which determine if an organism is indeed alive, we know that organisms must do all 7 of these characteristics. The answer is no. Viruses are not living organisms because they really can't do all 7 characteristics in and of themselves. They can't do what they do without getting inside a host cell, a living host cell. And then what they do is they pirate the host cell, specifically the DNA of the host cell, in order to have that cell actually do its function in relation to the virus itself. So really, they do not do any of the characteristics of life, so they're not alive. All right, let us take a look at the relative size of a virus, just so we know what we're looking at as far as comparative to a cell of different types. So microscopic organisms are measured in 2 types of measurements. They're measured in micrometers and nanometers. A micrometer is 1/1000th of a millimeter, and a nanometer is 1/1000th of a micrometer. So nanometers are smaller than micrometers. Typically, when we're looking at things under the microscope, we measure things in micrometers because usually we're able to see a cell in micrometer measurements. So a typical plant cell would be about 100 micrometers, give or take a little bit. A bacteria cell is relatively smaller than a normal, let's say, plant cell, but you can still see them under the microscope. And you could measure them also in micrometers. They're about 1 micrometer, which is 1,000 nanometers. Now, a virus, a typical virus like the spherical virus that we talked about before, we're talking about 100 nanometers. So that is 1/1000th the size of a typical plant cell. So a virus is much, much smaller than a typical plant cell, much smaller than a bacteria cell, smaller than even some organelles that exist inside plant and animal cells. And this organelle that you see here is a mitochondria. So the fact is that they're much, much smaller, so that makes it much easier for them to be able to get inside a living cell. So I just wanted to point that out so we know kind of what the size of what we're talking about here is. Much, much smaller than a living cell, much harder to see under a microscope. You have to use extremely powerful microscopes in order to actually see a virus itself. All right, so let's get into the structure of a virus. So we're just going to look at a typical virus, in this case, a spherical shaped virus. All viruses will have the same types of features, so it doesn't matter which shape you're looking at. Generally, there are three basic parts, sometimes just two, but because we are looking at the spherical virus, which again is an example of like an influenza or a COVID type virus, these are the structures that the virus will have. So first of all, we're looking at what's called the virus is surrounded by a protein coat called a capsid. This is not a cell wall. It is just the outside container, so to speak, of the virus structure, and it is made of a protein. It's called a capsid or a protein coat. So the second structure is genetic material, and that is housed inside the protein coat. Now, the genetic material of a virus will either be DNA, which is a double-stranded genetic code, or RNA, which is a single-stranded genetic code. It just is the type of genetic material that the specific virus would have. And when virologists are studying the virus, depending on whether it's a DNA virus or an RNA virus, then they will have different modalities of dealing with that particular virus. The third structure, which you would find on a spherical shaped virus, which may not always be found on every virus, are these proteins that stick out of the protein coat. So it is part of the capsid or the protein coat structure. But they are, I would say, like appendages that stick out, that these specific proteins allow the virus to actually get into a host cell much easier. These proteins that stick out have different shapes, and what happens is it's kind of like a key that fits into a lock, and the lock is on the host cell. So if the protein is the right shape or the right key, it will be able to access the lock on the host cell. And when it fits in, the host cell will allow the virus in because of the shape of the key that the virus has on it. So that's how a virus can easily get into a host cell, any kind of host cell. So that's the structure of the virus, just the three parts. It's got the protein coat, also known as the capsid, some type of genetic material. When you're labeling the virus, you want to say genetic material. You also could say DNA or RNA. Do not just put DNA. Do not just put RNA. You have to include both of them because you don't really know which one it is in the diagram. And then these little appendages that stick out, if your diagram has them, those would just be protein structures.